So here we have the actual process of cellular division depicted in figure 1012. Right, figure 1012? Sorry, 1011. Figure 1011. So we start off with the interface. At the end of interface, the G2 phase. Okay, and this leads us into the actual stages within mitosis. We have prophase, and in prophase, here we have the condensation of our chromosomes. So we have the actual chromosomes becoming visible at this point in the cell cycle. You have also the spindle fibers being formed and the centrioles moving to opposite poles. Okay, as you can see here, they're starting to move to opposite poles. You also have the disappearance of the Golgi and the endoplasmic reticulum, the major components of the endomembrane system, along with the breaking down of the actual nuclear envelope, okay, which is also part of the endomembrane system. Whatever begins in prophase actually ends in prometaphase, as we can see here. And that's basically what the distinction between prophase and prometaphase is. It's just prophase begins it, prometaphase ends it. Okay. You have the spindle fibers which reach the opposite poles. The centrioles which reach opposite poles, and you have the formation, the near complete formation of your spindle fibers, which are very close to connecting to the kinetic pores, which are located on the centromeres, on the chromosomes. And these chromosomes, which have now fully condensed, are moving towards the metaphase plate, the equatorial plate of the cell, to proceed on to the next stage of cellular division, which is metaphase. With metaphase, you have the lining up of the chromosomes at the center of the cell, at the metaphase plate or equatorial plate. Okay? And at this point, you also have the attachment of your spindle fibers onto your kinetic pores. Okay? Let's see. And from there, we have anaphase. Anaphase is where we actually have the chromosomes being pulled apart, as you can see there. So the chromosomes are being pulled apart, okay, and pulled in opposite directions towards the opposite poles. And this is done by essentially shortening the strands of the spindle fibers. Once all the spindle fibers have attached onto the kinetic pores of all the chromosomes, and you have the separation of these chromosomes, and it does happen essentially randomly, which leads us into the law of segregation a little bit later on. Okay, so once we get into the law of segregation, it's very important that we make this connection back to this point in this chapter. Okay, and that comes along in chapter 13 next week. Now, anaphase, again, you have the pulling apart of the chromosomes, and then in telophase, that's where you have the majority of the elongation of the cell, even though you do begin elongating the cell during the second or the latter half of anaphase. Okay. Now in telophase, you continue the process, the elongation process, and you actually begin the reformation of the endomembrane system. You essentially do you redo everything that prophase undid. Okay. The reformation of the nuclear envelope, the decondensation of the chromosomes, the deterioration of your spindle fibers. You have the reformation of the rest of the endomembrane system, the Golgi and the ER, amongst other things that all happen within telophase. And so telophase moves us into the process of cytokinesis. And with cytokinesis, you literally have the division between the cells. Okay, the divisioning of the actual cytoplasm between the cells, as you can see here. We've already formed the two separate nuclei, amongst all the other steps that reformation steps that happen during telophase. And here in animal cells, we actually form the cleavage furrow, which we can see depicted here. They're in a stained cellular form. And here we have the cleavage furrow as well here in two separate micrograph depictions. Okay, that is the cleavage furrow that happens in animal cells. But in plant cells, it happens slightly differently. So with plant cells, it actually, the vesicles that are formed via the use of the Golgi make up the cell plate, okay, or will make up the cell plate that will form the division between the two newly formed plant cells. Okay. Now in the next video, we'll actually talk about the control of this cell cycle.